Okay, I think we've got a reasonable audience, so we might as well get started. Tēnā koutou katoa. Thank you for joining us this afternoon on what is probably a rainy afternoon for most of us. Just a quick apology for those of you that tuned in to our last seminar that I hosted, and I neglected to introduce myself. So for those of you who don't know me, ko Tracy Godfrey ahau, he uri ahau no Ngāti Awa, Ngāti Tūwharetō, Ngāi Tūhoi, Ngāti Maniapoto, Ngāti Maru, Ngāti He Kairo. And I take care of vision mātauranga aspects for Genomics Aotearoa. So getting on to today, we have Keith Funnell and Wayne Blissett joining us. And they're going to be talking about a project that Genomics Aotearoa supported through our Indigenous Genomics platform. And I guess you can all see the presentation is already on your screens. And I think, actually, I probably don't need to explain anymore because I know that they're going to cover everything we need to know in their presentation. Feel free to pop any pātai, any questions into the chat, and following the presentation, we will get some kōrero going with Keith and Wayne about this great work that they've done. So thank you both for taking this time out for us, and I'll hand over to you.
Kia ora mai no tātou, kani te me ki aku I Tracy, mo tēnei mihi. Kia koutou mā, nā kaiti aku o te pūtaiao, ko tēnei te mihi nui kia koutou, kua mahi a te wā he whakawātia te wā ki te kōrero tia tātou nei huatahi, koini e te rohe o Rangitāne o Manawatū, o no reira no te aroha, no te Māori o te whenua, ka mihi a te kia koutou katoa. So I'm Wayne Blissett, originally from the beautiful Winterless North, but currently working under the mantle of Tāne Nui a Rangi here in the mighty Manawatū. My role is as the Executive Director of Tui Aotearoa, and we have our confirmed Tiriti partnership with Plant and Food Research, working on a number of different scientific endeavours, and this one is a particular passion of ours. So I'll pass over to Keith. Kia ora rā.
Tēnā katoa. So Keith Funnell, based in Papioia as well, and I will simply move on to the title of Weaving of Indigenous Māori Knowledge and Science, and the focus is realising the potential of genomics for Swamp Māori. As Wayne just noted, there is a partnership that exists between Plant and Food Research and Rangitāne o Manawatū. And so I'll start by just giving a bit of a one-pager synopsis of that partnership. So we've actually been working together at some level for over 20 years. Initially, of course, it was that token gesture, initially regulatory requirements when dealing with genetically modified organisms or human health trials. But since 2018, we've actually been undertaking a lot more joint projects, like looking at karaka as a taonga food, and the potential of creating an enterprise out of that with Rangitāne o Manawatū. Looking at urbanisation of runga rākau, so medicinal plants, and how we might be able to better utilise them within our urbanised environments. Also looking at Swamp Māori, its conservation and restoration, which is the central focus of today's presentation. But in addition to actually looking at individual rākau species like karaka, kumaraho, Swamp Māori, we also have been doing in partnership things at perhaps a higher level, presenting internationally about ethical plant access for plant breeding and use of taonga species, cultural confidence training of our PFR staff, and perhaps just provoking Wayne a little bit, I don't know, by saying perhaps it was actually a two-way street of Rangitāne o Manawatū folk learning about the culture of our pūtaiao science system as well. a lot for both parties to learn, I think, in the partnership. Multiple, yeah, no, that wasn't an invitation for you to say anything, Wayne, no. So multiple joint projects, a diverse array of topics. And when we get to the question and answer discussion section, you know, I'm hoping you're asking questions about what makes the partnership work. So let's start by jumping into the Swamp Māori program, and let's look at the background to that. And just fortuitously in this month, earlier on, the Taonga or Manawatū highway opened for traffic, and this forms the focus of our first bit of kōrero. Now, hopefully a brief video is playing on your screen. Describing the 11.5 kilometre long highway from the west coast to the east coast, involving obviously multiple iwi partners in the construction and its development. And hopefully I am going to go back now. If you recollect in that brief video, right at the beginning, there were a number of bridges. One, of course, crossing the Manawatū River itself. And here we see in one of the flyover video images that Transit New Zealand have published, bridge number 3, known as Echo Viaduct. So to the right of where that bridge is labelled Echo Viaduct, we see a bit of a ravine or a viaduct with a lot of regenerating bush to the right, and to the left we had cows and goats and deer and all sorts of things grazing up until a couple of years ago. And here in the red circle is our little swamp area with what we calculated about 20 remaining trees of Swamp Māori were left in this Echo Viaduct. So as a member of this multi-iwi collective working with government, etc., Rangitāne or Manawatū, or ROM as we affectionately call them, made it quite clear. They came to us as plant and food research to say, we need a conservation plan, a restoration plan of some sort. And we got to that point with some kōrero to say, let's weave our mātauranga Māori and pūtaiao together to try and address this problematic situation. So here we are under bridge number 3 in its early stages of construction. So those steel girders in the left-hand image going over there, and Paul Horton from Rangitāne o Manawatū's Te Aotearoa or environmental team assisting Colon, who was undertaking a PhD, and depict there in the alongside Paul, sampling plant material for DNA analysis so that we can quantify the genetic diversity of the Swamp Māori that were left within the rohe. And as you'll see in forthcoming slides, Rangitāne gifted the genome a name. Oh, Wayne, do you want to jump in and do good in pronunciation of our reo?
Kia ora, Keith. Yeah, we went through quite a process through that iwi kaupapa to gift a name that carried the significance of the kaupapa that we were undertaking of Nahua or Ia Whenua, translated loosely the fruits of the land in regards to understanding the role that the genome sequence would have moving forward.
And the tree that Paul in his orange vest is got his left arm out to on the right-hand image, that is what was affectionately known as plant number 6 initially before it got its name. And then we put Te Matapihi in front of the genome name. So the literal translation is the window of, or window to, or through to the fruits of the land. So it reflects, we've named the plant from which has given us these genomic insights into Swamp Māori. and perhaps acknowledges the fact that swamp Māori occupies whenua beyond the rohe of Rangitāne on Manawatū. So it's just a window through to so much knowledge. So for those of us that are perhaps panting and wanting to know all the exciting genomic information that was derived, here's just a small snippet of it, a principal components analysis of that genetic relationship between just the plant material involved in this project, which was some trees that we sampled from the eco viaduct, which we just looked at, the Ashes domain, which is just down the road within the rohe, and also Ferry Reserve. So there were 3 last remaining remnant populations, and also we had some seedlings that we had established of each of those. So we can see there through that principal components analysis, there's some genetic diversity there, and obviously any good restoration conservation plan wants to capture as much of that genetic diversity as possible. Just subtly sitting there on the right hand side bottom corner of that image are our use of biocultural labels. So Rangitāne has agreed and has signed up to the local context hub, and so has plant and food research. So in our collaborative partnership environment, as we create new knowledge, new data, we apply the biocultural notices, biocultural labels, indicating the, I suppose, respecting the kaitiaki role of Rangitāne o Manawatū in undertaking this mahi, and also going forward with that data. So also the work comprised identifying sites as part of the restoration plan where we could do plantings. And you'll just see there in quite simple terms, 5 sites for restoration were identified using Lucy, which was a platform of using hydrological data, rainfall data, soil profiles, etc., and computer-generated decision-making to work out perhaps sites where we could plant swamp Māori, because it does really need quite swampy ground. In addition, 3 sites were identified using Indigenous knowledge, mātauranga Māori. In our working relationship, again and again, I'm always put back in my place when our partners will often say to us, "Thank you, Keith, for telling us what we already knew," as if we needed to tell our partner what a swamp looked like. But anyway, we were able to get some coordination. You'll see in Blind Island and Mautoa floodway, there's an alignment between the red and blue dots, which says we agreed that this was a suitable swampy area to plant swamp Māori in. But the red dots also reflected more sensible logistical issues of these were, the red dots were areas where Rangitāne o Manawatū already had working operational sort of activities in those swamp areas where they could readily begin implementing the restoration plan. Wayne, do you want to take the talking stick here?
Sure, thanks, Keith. I think the fundamental framework for our working relationship with Genomics Aotearoa and plant and food research is based on the Y262 best principles. And you'll see there at the bottom the 8 principles and values that we've taken from the guidelines there on the right. And we've translated them to make them practical and workable for both of us as organisations. And looking at kotahitanga is the importance of a shared understanding that honours tiriti and builds mutual respect between both parties. Kaitiakitanga being the recognition of mana whenua as kaitiaki of taonga species and the duties that incurs for mana whenua. And tino rangatiratanga looking at the recognition of kaitiaki as an inalienable right that is guaranteed through tiriti, and how it is that we work through access and benefit sharing as a process of tino rangatiratanga and mutual respect. And exploring also through whakawhanaunga and making sure that we develop respectful relationships, and it's not a right-wrong conversation so much as a difference of understanding the world. And it is having those strong relationships to talk through the difficult questions or issues that may come up, and particularly continuing to build the collaboration opportunities. Importantly also is kia tūpato, and I think that's the one we often struggle with each other, probably, to be fair. And that's the need to take things a little bit slowly and consider things rather than rushing through and getting things done because we want to get it done. But actually taking the time to reflect and have the time to have the conversation with each other, so there is that shared understanding when working with taonga together. Te ukai pōtanga is really just reflecting on, as Māori as first beneficiaries of taonga species as a kaitiaki, how do we translate that in a way that recognises the contribution of both pūtaiao and mātauranga to what could be new IP or existing IP, and having a serious kind of reflective moment on how it is that we make sense of that new IP. Manaakitanga is, of course, our mutual respect, and that each of us as partners have mana that we bring to the conversation, and ensuring that we maintain a mana uplifting and meaningful kōrero. Even when things might be a bit tough, it's actually talking through that and making sure that we do it with mutual respect. And finally, te reo, ensuring that when we use te reo, we're using it correctly, and that it's in context and used in the right way to be able to whakamana the whakapapa of the taonga. So to give the taonga its deserved respect and place, and how it is and why it is that we talk about it in such respect. Over to you, Keith.
In terms of the partnership itself, as the heading on this page sort of says, we have taken those principles and utilised them at regular intervals every year to assess our progress in aligning with each of those principles and values, and I suppose, as a means of self-checking how we're going. So that sort of summarises what we've done in the specifically in the Swamp Māori project, developing a restoration plan. I think it's in an environment where we're so often, wearing my CRI hat, we're so often in our relationship so contractual. Get a contract, get it signed, deliver the milestones, and then wash our hands, we're out of there. But I think it's important to recognise that we've got especially with working with mana whenua, we've got a long-term commitment, and we see how this needs to perhaps be supported by perhaps considering the ripple effects of each of our bit of mahi. And so one of the ripple effects, of course, was, we published the genome, and that was a world first. We believe that when Rangitāne or Manawatū gifted the name in te reo for a new genome, and that is now the genome is housed in Genomics Aotearoa's AGDR, and which ROM is kaitiaki over that data again, as it is for the Manuka data, and for soon we hope with genome for Karaka as well. And of course, not forgetting Colin here got his PhD awarded as well as a result of that. So some wonderfully powerful ripple effects from that mahi. But also at the same time, there were people who weren't involved in that particular project per se. So we developed and refined the vegetative propagation of swamp Māori, which actually isn't as easy as you might perhaps suggest, that it's a simple horticultural practice, but getting it up to 90–100% strike rate of cuttings was a bit of a challenge, and so we've been able to also publish that underlying pūtaiao or science associated with that, and we know that's within our academic environments, that's important to us. Rangitāne folk perhaps yawn a little bit, but we know that's the taskmaster that we beat to. So what was the point of doing a restoration plan unless you're actually going to implement that plan? So these images here collectively show members of Rangitāne or Manawatū Te Aotearoa team, so their environmental team, undertaking the plantings in swamps of plant material that we were able to propagate and knowingly plant that plant material with the greatest genomic diversity at each of the sites where we were planting. So illustrating a very powerful use of genomic information and turning it into real life practical outcomes, I believe. Also in this image here, we're just illustrating that plants of the known genomic profile have been then used elsewhere within Plant and Foods program of work with Swamp Māori looking at myrtle rust. So here's Hanarera taking charge of some newly propagated plants for undertaking that work, but also illustrating the next generation of ROM workers coming in through the system that are being exposed to that weaving of mātauranga Māori and pūtaiao. And also, I think if perhaps I'll hand the talking stick to Wayne again here as well, because that the impact that has had on members of the Rangitāne members, and also this last bullet point here about increased awareness that use of the word genes and gene research doesn't have to conjure
Sure, thanks, Keith. I think the fundamental framework for our working relationship with Genomics Aotearoa and plant and food research is based on the Y262 best principles. And you'll see there at the bottom the 8 principles and values that we've taken from the guidelines there on the right. And we've translated them to make them practical and workable for both of us as organisations, and looking at kotahitanga is the importance of a shared understanding that honours te reti and builds mutual respect between both parties. Kaitiakitanga being the recognition of mana whenua as kaitiaki of taonga species and the duties that incurs for mana whenua. And tino rangatiratanga looking at the recognition of kaitiaki as an inalienable right that is guaranteed through te reti, and how it is that we work through access and benefit sharing as a process of tino rangatiratanga and mutual respect. And exploring also through whakawhanaunga and making sure that we develop respectful relationships, and it's not a right-wrong conversation so much as a difference of understanding the world. And it is having those strong relationships to talk through the difficult questions or issues that may come up, and particularly continuing to build the collaboration opportunities. Importantly also is kia tūpato, and I think that's the one we often struggle with each other, probably, to be fair, and that's the need to take things a little bit slowly and consider things rather than rushing through and getting things done because we want to get it done, but actually taking the time to reflect and have the time to have the conversation with each other, so there is that shared understanding when working with taonga together. Te ukai pōtanga is really just reflecting on, as Māori as first beneficiaries of taonga species as a kaitiaki, how do we translate that in a way that recognises the contribution of both pūtaiao and mātauranga to what could be new IP or existing IP, and having a serious kind of reflective moment on how it is that we make sense of that new IP. Manaakitanga is, of course, you know, our mutual respect, and that each of us as partners have mana that we bring to the conversation. and ensuring that we maintain a mana uplifting and meaningful kōrero. Even when things might be a bit tough, it's actually talking through that and making sure that we do it with mutual respect. And finally, te reo, ensuring that when we use te reo, we're using it correctly and that it's in context and used in the right way to be able to whakamana the whakapapa of the taonga. So to give the taonga its deserved respect and place and how it is and why it is that we talk about it in such respect. Over to you, Keith.
horror stories and be controversial. Wayne, do you have any little add-on through that?
Yeah, I think you've captured the essence. And I guess I was having a conversation with another CRI yesterday around benefit sharing, and used this example exactly when Rangitāne Manawatū was noted for being possibly the first Indigenous people to name a genome. It actually gave a profile for our descendants and beneficiaries to see science as a thing that the iwi was involved in, and that there was a career pathway there where they could follow their mātauranga Māori or pūtaiao or both. And it changes the discourse of, oh, that's something over there, as opposed to now in the iwi, it's a consistent conversation about working with Genomics Aotearoa and plant and food research, and understanding that science is part of our solution collectively as a nation and as a community. And similarly with our rongoa corridors, working to put rongoa and taonga species throughout the urban ngahere. Once again, our beneficiaries and descendants are seeing how it is the partnerships and collaborations help deliver the aspirations that we hold collectively as a community.
And if you can see on this image here, on the right-hand side, tilting off to one side, is part of an excerpt of one of Rangitāne o Manawatū's annual reports, and there's specific mention being made there in the 23-24 about Genomics Aotearoa, karaka, swamp maire, and a range of the different projects that and reports that have been delivered together with Rangitāne, not to Rangitāne per se, but together with, and because you'll see Wayne's name as a co-author on the report that's cover pages on the front there. So through that, I think we've generated a lot of awareness and change within the folk of Rangitāne o Manawatū to appreciate pūtaiao, and I know quite definitely the respect goes the other way as well. So our last slide, te tangata, te tangata, te tangata. We can't do this without many people being involved. So just acknowledging a list of people within Rangitāne o Manawatū, so the Te Aotearoa team, particularly Paul and Renata, Alana and Mel, Victoria University, Colin and Julie, who assisted in the developing the restoration plan, and then within Plant and Food Research, David Chanier, Emily, Stephen, Belinda, Donna and Andrew, as well as acknowledging our sponsors, Genomics Aotearoa, of course, Rangitāne and Plant and Food, Victoria University, and of course the National Science Challenge, which helped fund some of the work associated with Colin's PhD as well. So that gives a brief overview of our activities and introduces what we hope to present today. But I hope what we presented today provokes some questions for you to consider. So I'm going to stop sharing.
Ngā mihi kia kōrua, thank you very much for your presentation. Feel free, audience, to pop any pātai that you have into the chat, and in the absence of questions at the moment, I might just get us started with a general question in terms of the relationship between your two organisations, between Plant and Food and Rangitāne o Manawatū. It appears to be through this project and the other projects that you've engaged and worked together in, that you have quite a strong and respectful collaboration. I wonder whether it has always been this way, given the culture of research institutions in the past. So question to you both, has the relationship always been this good? Perhaps it has, I'm not sure. But also, do you have any insights that you can share with other researchers, given that you're both on opposite but complementary sides of the fence, so to speak?
Well, it's Wayne's going to talk about that, aren't you?
I thought you were, because you got more of the historical analysis, but I think from what I understand, probably as Keith did kind of highlight at the beginning, the relationship traditionally was probably far more didactic, tick box exercise that probably people were more used to, particularly, I guess, in regards to getting approvals, for lack of a better word, from the iwi, and not having actually a consistent approach to how that might have been undertaken. It could have been this person or that person, so long as they were a part of the iwi, then that was okay. And I think what's really supported the relationship to become more robust is having tangible projects and a commitment to a treaty-based relationship where there's a structure, a form, and a practice that's required by both parties to achieve some shared outcomes and aims. Rather than a casual kind of ad hoc, we've been very deliberate in creating a systemized approach to how it is that we do things between us as partners. And I think that's been important to create clarity for both parties about what the expectations are and what the behaviours are that allow the best practice model to emerge between the two of us.
Congratulations to you both, and in having that willingness and good intent on both sides of the partnership. It's great to see. We've got a comment and a question here from Damien, who says, love to see the experimental design factors for the propagation phase of the project, and were any suggested by the partners?
Well, that was done, the actual development of optimizing the propagation was outside of the Genomics Aotearoa funded activity, but the peer-reviewed paper that was published on propagation is available, and I can, give the link to that if required. But we went on and refined that even to optimize it even higher. But basically looking at the stage of growth of the plant material, how active the plants were, and also using plant growth regulators to determine best ways of actually getting rooting to occur. So I can provide that link.
Thank you, Keith. Grace says, tēnā korua mō o kōrero, interested in the IP and data sovereignty aspect. I think Keith mentioned that Rangitāne or Manawatū are the kaitiaki of the genome data. How does this play out in a practical sense in terms of where data is housed and how decisions are made regarding data use by scientists? I'm just thinking, Wayne, it's probably good for you to speak to this, but I can also add something, and Claire would probably like to add something as well, but if you'd like to go first, Wayne.
Well, so I guess the easiest response, which I think is why Chase has thrown it my way, is that it sits on the AGDR. So it's housed by Genomics Aotearoa. So for those that want to access the genome sequence, there's an application process to go through. And as kaitiaki, that's forwarded to Rangitāne or Manawatū to review, to look through how the genome sequence would be used, the deliverables, the connections back to Māori partners, and from that we'll make an assessment as to whether people get access, and we'll return that back to Genomics Aotearoa for processing.
Kia ora, Wayne. Claire, would you like to jump in, and if there's anything I can add, I'll add after you, but between the two of you may cover it all off.
Yeah, thank you. Just so I guess I work with the AGDR, so just to reinforce what Wayne said, we're the in-between, I guess, the messenger between any researchers who want to use the data and the kaitiaki for that data set. And the data itself and associated metadata is all housed within New Zealand, so it remains the data sovereignty within New Zealand, and so it's hosted on NESI's national platforms. And as Wayne said, the access is only granted when kaitiaki have approved, and there's additional terms that can be added to sort of the standard terms around use and any benefit sharing or anything like that is felt should be needed. And where appropriate, we ensure that data doesn't actually leave New Zealand. And in some cases, we have had data sets that are okay to be used overseas, but that's very much absolutely in the decision of the kaitiaki.
And it'd be fair to say, Claire, that sometimes the answer has been no for access?
Absolutely. Yep, that's a very good point. So there's plenty of examples, I guess, especially with overseas applications where maybe culturally there's not so much understanding of why the data set is controlled access. And so often there's not a lot of details. It's very sparsely, the form is very sparsely filled in. We won't pass that on directly to kaitiaki. We'll work with the researcher to try and complete and give a full picture of what research they're trying to do. Often at that point, especially if there's sort of commercial aspects, we lose touch. They stop answering us. But in kind of the positive outcomes, we can work with them to get a complete application form to pass on to kaitiaki. But both at that, can we get a complete form, and at the kaitiaki assessment stage, we have had applications denied.
Probably all I can think of to add to that is, just for people that don't know about our Aotearoa Genomics data repository, that we strongly support the principles of Māori and Indigenous data sovereignty. Obviously, in an ideal world, the hapu or iwi themselves would have their own server and be holding that genomic data themselves. We're not in an ideal world, but we do strive to strongly support Māori data sovereignty through the fact that we have a kaitiaki over each data set. And probably just the other thing is that kaitiaki have the ability to impose certain conditions, so they may give permission. We have a general default position that kaitiaki agree to, but they may want more stringent conditions, or perhaps in the future, if there's application that has commercial benefit, kaitiaki, that would be their choice if they wish to actually allow that, if they were involved in or if they were able to derive commercial benefit as well. So hopefully we've covered Grace's question. And we've got one from Anthea who says, Forest and Bird, along with DOC and QE Trust, managed to get NZTA to reduce the size of the footprint over the seepage swamp because of the swamp maire. And I have since learnt no swamp maire were lost by building the Eco Bridge 3. That's sorry, not a question, but a comment, but that's good to hear. And we have, somebody has kindly posted the link for, Georgina has posted the AGDR link, if anybody would like to check that out. I just had a question around, obviously the low numbers of swamp maire and rohe are concerning, but I guess even more concerning given the myrtle rust threat. Had that, Keith, you mentioned that there's other research going on at PFR regarding myrtle rust. Has that had any results come through in terms of swamp maire, or?
At this stage, I'm not aware of it, but I'm not central to the diverse array of activities. But there was a lot of stuff under the National Science Challenge, which is being wrapped up. So some of that was happening in Papioia was on trying different methods of cryopreservation of the genetic material was one of the strategies. But Handarea's project is looking at the microbiome of the microflora associated with our swamp maire in different geographic locations, and whether there are any golden threads there, but I don't know what understandings have been developed from that as yet.
Right. And just another myrtle rust question. Is myrtle rust established in the area on affecting other species? Or...
Don't say that.
Sorry.
Uniquely, gosh, what a terrible conversation to have to say. But yeah, we have had incidents of it, but our swamp Māori and the rohe seemed relatively unaffected compared with what we see and hear from elsewhere in the motu. So maybe the genetic robustness is so concentrated and unique that ours are naturally resistant.
That would be great.
That would be great.
In terms of naming the genome, I thought that was heartening to see. Wayne, with the Manuka genome that Rangitāne or Manawatū is kaitaki of, is there a possibility to name that also, and with the upcoming karaka?
I guess anything's possible. Absolutely. I guess that it is an important process and understanding the whakapapa and the context to how the genome came to be. And I guess with the Swamp Māori one, we knew its whole whakapapa and background because it was done right here, right now with us. Whereas the Manuka genome is a more historic genome in the database. And so we would, work to find a way through to be honourable to that history and, find an ingoa that would suit the uniqueness of that genome experience. And similarly with the karaka, because we've been a part of that entire process, then yes, if the opportunity came forth, then we would be happy to have that kōrero.
If it was appropriate, and since we've got Claire online, is that something that's easily done in terms of changing the names of the genomes that we already have stored in the AGDR?
Yeah, we could certainly work to update those names. Yeah, absolutely.
Got another comment here. Since or while developing this data, did you identify other or new species of plant life that could be part of future joint projects?
I know when we were sampling in Eco Viaduct, Paul was getting me to take photographs of plants that were nearby as part of the ecosystem that he was intrigued by. He hadn't seen large leaf forms of this species or that species. But it wasn't, we didn't set out to do a complete ecological transcript of what was present in each of those environments. So we have a list of plants together with Rangitāne or Manawatū that we have are developing a shared research agenda about, and that we will be taking those forward. That's cognizant of resourcing requirements, but yeah.
In terms of the restoration sites, and I know you touched on this, Keith, in the presentation, for the pūtaiao or science-based restoration sites, could you just clarify what were the biological or ecological criteria for selecting those science-based sites?
I don't know them off by heart, but there is a complex array of hydrological soil type data, rainfall data, etc., that was all added in, flood proneness, which have been that Julie Gillespie from Victoria University and her team have been utilising conservation of swamp Māori in the wider Wellington region. So they had already test driven and developed and refined it based on those projects. And so it was just a matter of rolling it out and applying it to the rohe of that we were involved in.
Right. Wayne, in terms of the 3 sites that were selected by iwi, are you able to share the cultural criteria or mātauranga behind those sites?
Yeah, so a number of them were within the Rangitāne or Manawatū whenua returned through the treaty settlement process, and they're all within the area of interest of Rangitāne or Manawatū. So we also know that they were swamps, which was our criterion. You know, at least not overthink it, but anyway. And of course, with the Te Aotearoa team, they're out looking after our cultural properties as a part of their scope of practice. So they have a good knowledge of the whenua and where those, I guess, the same kind of criterion when you look at the flood, how long did it stay wet in summer, some of those criterion that help succeed or help swamp Māori to succeed in that environment. So while it was mātauranga Māori, it was about knowledge of the whenua and the intimacy with the land, but the principles and the components were not dissimilar. And I think that was a really good example of where mātauranga and pūtaiao don't fight, they coalesce beautifully when you actually take the time and sit back rather than trying to force one or the other. If you can just sit back and let both of them come forth, they actually do weave together beautifully. And I think it was a really good example of how that can happen. I think the other thing I would add is actually for plant and food research in Rangitāne, like we got this gig, we're pretty good. We work at, you know, we have our little fights, our little disagreements every now and again. But this one pushed us also invite Victoria University, you know, and have another party and another conversation and making sure that actually our rules of engagement were kind of the rules of engagement. So I think that's another interesting thing moving forward when iwi and mana whenua and these partnerships and other parties come in through a research process, how that process of engagement is undertaken about the rules, the principles and the practices of how we do things here is really important, so that I guess there's a shared understanding of how this cope-up is going to work. Because it took a little bit of time, but they got the gig eventually. But it is actually being able to take others on that journey and about, you know, these are our best practice principles and working together and taking others on that journey, I think was a really important bit. So that kia tūpato came in as kind of a key value while bringing in another partner to the kōrero.
I was going to ask, but you've kind of just answered it, Wayne, but I'll sort of ask anyway in case you either of you have anything to add. But I did wonder whether the relationship, the collaboration that you have, whether you feel that you're all the exception to the rule, or whether you're heartened that this sort of thing is going on everywhere. But given that, you know, different institutions, different researchers are at different levels, I'm thinking perhaps you might be, your relationship may not be common across research institutions, which I think is sort of what you were just alluding to, Wayne. But yeah, I don't know if Keith, you obviously work well with Rangitāne or Manawatū. Do you feel that it's an institutional thing for your institution, or that everyone is still in an earlier learning phase?
I think we all never stop learning. So I know myself, I've got plenty more learning to do. But even as an institution, plant and food research is not perfect. Not every individual is necessarily at the same point in their journey. And so that's what we've been finding with projects that then take on a new aspect, and we start bringing in other people from other locations from outside the rohe that are plant and food employees. It takes a while to bring them into acknowledging and accepting and operating within the values that the Y262 document sort of sets out. So yeah, certainly it's not just me, but it's wider than me within plant and food, but I acknowledge it's not everyone. And outside plant and food, again, there's another array of individuals. So I think Aotearoa in general is well placed internationally to stand up and be proud that we are taking steps and leading, I think, internationally, some of this sort of working with Indigenous peoples and in a respectful way.
And I think it's great that you share your experience of working together, because there's often good intent in the research community, but people are just not sure how to go about things. So by sharing your experience, hopefully it helps steer people in the right direction. If we have any more pātai from our audience, I would encourage you to post it now, because I'm thinking without any more questions, we can possibly have a slightly earlier finish. Both of you, Keith and Wayne, is there anything that wasn't mentioned in the presentation, a key learning that you have from working together with each other, or from this specific research project that you'd like to share, or do you feel you've covered it all in the presentation?
Haven't even scratched the surface.
Oh,
The reality is that it's so complex and so deep that what we've presented today is a nice, pretty bounce across the top example of one of our activities. So it's a bit hard to know what else to offer at this point right here now.
did you have anything to add, Wayne? Or?
Yeah, I think I would just like to acknowledge plant and food research and Genomics Aotearoa to that end, through having meaningful conversations and wrestling with some of the tough stuff and changing maybe the old practices and being brave to have courageous conversations of working with mana whenua in a meaningful way. Given it is a new, or not new, but a challenging environment, I guess, across all of science at the moment. It's also the opportunity, I guess, to be able to strengthen those partnerships with mana whenua so that together we can create a best practice model that truly acknowledges Indigenous knowledge systems as a part of pūtaiao as a best practice model, not only for Aotearoa, as Keith said, but for the world, and how it is that we can use traditional understandings alongside breaking technology and scientific techniques to really create, I know the bioeconomy is a big thing at the moment, but innovations and opportunities for our mokopuna to come. So I acknowledge plant and food research and Genomics Aotearoa, and also, I guess, just preparing ourselves for the digital partnerships that are going to emerge in the very short term in regards to how we formulate digital partnerships that are respectful and concerned about wise stewardship of data and taking that stewardship as a serious commitment, because that starts right from the very beginning of how data is first recorded and then how it's stored and then how the algorithm links it back to kaitiakitanga. So I think there's a new emerging discipline in regards to data digital partnerships, which I look forward to exploring in the future. So Māori ora ki te kaupapa.
That's a great point in terms of the digital advancements and where AI is taking us, and it's difficult for a lot of mana whenua to keep up with changing technology and with the speed that it's changing. So it's good to hear that you've got your finger on the pulse there in the Manawatū. We have got something else. Another question has popped up in the chat from Damien. In terms of data sovereignty for Rangitāne or Manawatū, if the nearest plant and food research office can access the RIAN's high-speed data backbone already, is it feasible in the future to investigate the possibility that Rangitāne or Manawatū might one day hold the up-to-date genomic data at their own secure server farm site?
Yes, absolutely. And we're currently exploring what that can look like. And that's another project we're working with Plant and Food at the moment in regards to repatriating Rangitāne or Manawatū data out of plant and food research to our own data warehouse. So absolutely.
Fantastic to hear. Claire, did you have any comment there, given that that's in your sphere?
Yeah, I think just to echo really what Wayne's said, from an AGDR perspective, it's something that we've discussed and agree would love to do. And I think with I guess the way the setup is currently, there's no reason but funding, I guess, is the blocker here. I think from having data stored, within rohe rather than essentially at Nessie servers and well, now in Tamaki data center, because we've just moved. But I think long term that definitely is something that we would want to try and achieve, and hopefully, I think probably Rangitāne or Manawatū being a bit more mature than some of our other kaitiaki, this might be a prototype that we could look at relatively quickly, as opposed to some of the longer term plans.
Yeah, sure, clear. And I guess we are maturing in this space because it is something we're particularly interested in. So we also have health and social services. So that's also been part of our data conversation. I think we're also formulating our position and understanding data governance versus data sovereignty. So we would say with Genomics Aotearoa, we've got data governance as kaitiaki over the genomes. But if we were to repatriate it, we'd say we had data sovereignty. Because when you own and operate it, that's what we would call sovereignty. When you create permissions and access, that's when you've got governance. So that's how we as Rangitāne or Manawatū have made sense of the different stages and phases, I guess, on this data journey that we'll be going through. But also that both of those are clear commitments and expression of kaitiakitanga. So it doesn't have to be either, it can be both, and that's okay.
And I think definitely everyone involved with the AGDR is supportive of moving in that direction, where you and our other kaitiaki are able to have full data sovereignty rather than governance. What Simon says, ngā mihi nui. I think we can probably finish a few minutes early. Tēnei te mihi kia kōrua, thank you so much for your time and for sharing the project with us this afternoon. Thank you to our audience. Please, everybody, have a safe evening and weekend in this terrible weather. And we'll see you all again, hopefully in a fortnight's time.
Mauri ora ki te kaupapa, ka kite.